All right. Good morning, good afternoon, good evening, everyone, from wherever you're joining. Thanks for joining this event. My name is Radhika Bongoni, together with Patrick Koning, and also with the support of Formulations Association. We are organizing this webinar, Microbiome-Friendly Cosmetic Ingredients and Formulations. The agenda looks like about an hour and a half time frame with very interesting 4 speakers that we have and the topics. Broadly speaking, they cover different areas of the skin and also the microbiome friendliness around those areas. We have on the line first Paula Lisotto, product manager from BaseClear, and Sebastian Mongiat, head of global applications and customer support from DSM and B. Rob Howlin, microbiologist from GSK, Consumer Health, Surain Kierfleuf, sorry for the pronunciation, CEO of Lactobio. We intend to have the question session towards the end of the day. That's like after 5:10. So please leave your questions to the end. Very briefly about the organizing or host association or company, that's BaseClear. BaseClear is a CRO specialized in microbial genomics, has been established in 1995, so about 30 years of history, always in microbial genomics as a specialization. And it's a science-driven company, so publication is also a central theme to the organization. As an expert in microbial genomics and the service provider, we are specialized into different application areas. That's on microbial ingredients, that is from discovery to novel strains, novel enzymes, or even safety assessment for dossier filing with regulatory bodies. And then we have human health, which is specialized in gut health of an adult or of an infant, or also cohort studies, typically a partner for the clinical trials. Then we have skin health and personal care, which is specialized on the skin and various regions of the skin and microbiome as a link. That is mostly for dermatological, but also on the cosmetic sides of it. And we have animal health and performance, that's also towards the well-being of an animal and also association with microbiome. Think of sustainability projects like methane emission, or also as simple as probiotics for poultry, to antibiotic alternatives. So those are the kind of range of applications. And finally, the quality solutions, that's all about the quality control, vaccines, viral work around it. The organization itself is one-stop shop in its services, right from study design advice, wherein the experts join a project from our end, to sample collection, logistics, to analysis and data interpretation, again, towards solution that a client could use it for application or marketing or dossier filing. Also an accredited company for various certifications, as you see on the screen. This webinar is the format of an experts meeting that we do, experts meeting, which is a dedicated event with a very defined microbiome topic, with niche in itself, and also the experts speaking on throughout the day. And on the screen, you see logos of the past companies who have joined the experts meeting. And this webinar is a version of that experts meeting, but very specific for microbiome-friendly cosmetics and ingredients. Some house rules for today. There will be meeting highlights and summary that will follow up after the event. Please drop your questions only in the Q&A box of the meeting that you see. So there is a separate tab, please only drop there. If there are simple questions, which the speaker can decide themselves, they will answer them on the spot, which is via chat. And if it's something that deserves a discussion, I will put it on as a point to be discussed at the QA session that is in the panel. So please hold for that moment for discussion. And if there are still any unanswered questions, that we will pick with the speakers and come back to you after the event. With that, I would like to introduce our first speaker, Paula Lisotto. As mentioned, very interesting talk that she's going to give on measuring the microbiome, path towards products that are friendly to the microbiome. Enjoy the session, enjoy the event. Please make it very interactive as much possible. It's online, I'm aware, but I think we can do it with interactions. Thanks a lot. Enjoy. Paula, can you take over from here? Paula?
I'm doing it, yeah.
Perfect.
Yes. So do you also see the people on the right or just my monitor?
We see the screen.
Okay. Good morning. Good afternoon, everyone. My name is Paola Lisotto. I'm a product manager at BaseClear, and today I prepared a presentation for where I will discuss the steps that at BaseClear we recommend to study the skin microbiome. And these steps can lead toward, as the title of this presentation says, the development of personalized and skin-friendly cosmetics and therapies. Yes, so maybe I will give a very brief and short introduction on the skin microbiome. This is the second largest organ in the human body after the gut. And it has an area quite large of 1.5, almost 2 square meters. But the skin microbiome is not only doesn't work only as a physical barrier, but it's also an ecosystem which supports the growth of various microorganisms. In this ecosystem, we can find abiotic components, so non-living components, which dictate the environmental conditions and nutrients that can be found in the skin surface, but also biotic components, maybe the most known, which are characterized by the microorganisms and also the human host cells. Several studies in the skin microbiome have revealed topography and a topographical diversity in the skin bacterial communities, as presented in this figure. It has been identified several sites with a range of microbial diversity, from sebaceous sites with low diversity to, on the contrary, dry sites with a higher diversity. These studies are also consistent with the culturomics that have been conducted. So, but however, when it comes to skin microbiome, the analysis with next generation sequencing, as in this case, provided the highest resolution. At BaseClear, we have experience with different type of microbiome, with especially low biomass. This does not only include the skin, but also the scalp, where, for example, dandruff is associated with a disturbance in the ratio of bacteria and fungi. For example, the yeast Malassezia is known to be a cause of problems in this area. The oral microbiome is, for example, after the gut, the largest microbial community present in our body, and I'm sure we will hear more later during this webinar. The vaginal and the nasal and final the ocular are also microbiome with low biomass. And I have summarized in here some studies in which BaseClear has provided support in different topics. So as mentioned, we have experience with vaginal, skin, but also nasal and oral microbiome. So according to our experience, when it comes to study the skin microbiome, what we advise to do is to perform an in vivo study, so to perform a clinical trial. By performing this study, we evaluate together with our customers the product and the application areas. And afterwards, we define an optimal experimental strategy. This experimental strategy, of course, has to be based on specific characteristics that the study design has to keep in mind. Importantly, for example, the use of a healthy subject contrary to a more targeted group, depending on the type of product that wants to be used, and the ethnicity, because especially in cosmetics, some products are really focusing on specific area in the geography of the world. So we are able to work in a partnership with different CROs and ensure that the study can be performed in different type of populations. In order also to make sure that we are following the microbiome throughout its evolution and its dynamics, we always recommend to have multiple time points. And as any experimental strategy, we also recommend to have different type of controls, positive and negatives. So throughout this process, we, as BaseClear, we coordinate the clinical trial, we process the samples, and as a final result, we deliver a costume report. And here we summarized an example of a study design that we have performed in the past. Normally, as mentioned, we would advise to have a specific number of subjects, depending on the type of study, but especially because from a statistical point of view, it's important to reach significance, the higher the better, although we have specific standard, which I will come later to. We have dedicated protocol for skin collection and also for the transport of the samples. This is also a very important step in the process. Moreover, at BaseClear, we have developed dedicated databases to analyze the data that are generated from next generation sequencing. And from this data, we can then generate, as I mentioned, costume reports, which can be combined with advanced biostatistical analysis. And on top of it, additionally, multi-omics approach can be performed, which I will touch upon later. Another element that is really important when it comes to skin microbiome is to consider the fact that the skin microbiome has a low biomass. And this is also another element that is important to mention here is the high presence of the host DNA. For this reason, we have internally validated quality control methods that allow us to make sure that the samples and the DNA that we extracted from these samples is enough to proceed and obtain good results in order to make sure that, of course, the study can obtain the best final results. Moreover, as I mentioned before, at BaseClear, we have developed a specific taxonomic database which, as mentioned here, is represents more than 100,000 genomes, not only including common skin colonizer, but having also oral and vaginal genomes in it, and covering the whole diversity present in the body. So not only bacteria, but also viruses, eukaryotes, and so on. And of course, the fact that we have such a high diversity in our database allow us to have a high accuracy in the results to reach strain level identification, and especially to increase the percentage of classified reads especially when it comes to metagenomic analysis, are often a problem. So this is the whole flow chart of our services, and additionally, to the sequencing and the generation of costume report, we can complement our analysis with metabolomics, targeting both targeted and untargeted panels, having a wide range of analysis. We can also perform quantification of specific microorganism by targeting qPCR. We have already developed in-house a number of assays, but we are also open to develop assays that are picked by the customer. And we can also complement this with the more traditional tailored culturomics, basically growing skin colonizers and see which is the effect of the products on these microorganisms. But the part that I really want to draw your attention is the so-called microbiome tested report. So at the end of this process, BaseClear is able to provide a report where we are going to summarize the findings. And to maybe have more information about this report, what we do is, as I mentioned, use the information from the in-house dedicated database complement this information with possibly additional service like indeed the targeting the qPCR. And finally, analyzing the results, finding the relevance with the statistical analysis. And specifically, we can provide information at the taxonomic level per sample. We can look at the microbial diversity within the groups, but we can also perform more in-depth analysis, such as multivariate analysis, to investigate the beta diversity within the samples. Of course, additional more advanced analysis can be performed, such as machine learning and differential abundance analysis. And all this information can then be summarized in a final interpretation and conclusions of the study, which is going to be based on literature research and the safety of use of the microbial ingredients that are used in the testing. So in conclude, what we advise at BaseClear is to perform an in vivo analysis that at least would cover 3 time points in order to make sure we are aware of which is the baseline characteristics of the microbiome and how it evolved through time. This analysis is performed under strict coordination of a study director and is based on deep sequencing, which makes use of the dedicated database that I mentioned so far. All this analysis can be complemented with qPCR in order to target more specifically strains of interest. And if these criteria are met, then we are going to address and assess the reversibility of the impact of the formulation on the microbiome by, as I mentioned, investigating different measurements like alpha and beta diversity. And we are going to combine this information with advanced statistical analysis, considering the metadata such as the division in case and controls or different groups. So if overall the product does not disturb the commensal microbiome significantly, in this case, and also based, of course, on the interpretation of the literature research and safety of use, at BaseClear we can award a label, which is the microbiome-friendly label. In addition to these in vivo studies, we are also working towards the development of an in vitro testing, specifically targeting ingredients. For this purpose, we are going to generate in vitro testing, where skin mock communities will be used in different conditions, such as pH, temperature. These skin communities represent a common colonizer of the skin, representing different body areas. So depending on the area of application of the specific ingredients, we can create a different type of skin communities, and we can perform different type of assay. We aim to have this in vitro testing high throughput in order to make sure that several ingredients can be tested at the same time. And as previously indicated, we can provide for the in vitro testing as well, literature, research, and safety of use of the specific ingredients to make sure that these ingredients do not have an impact on the skin microbiome. So to conclude, at BaseClear, we can provide support in the starting a clinical trial, following up and investigating the effect of probiotic, postbiotics, and ingredients. We can do that by studying the skin microbiome using next generation sequencing and different omics techniques, including also targeting specific strain via qPCR. And as I mentioned, if the criteria of the ingredients or the formulation meets our internal standards, we can award the product the so-called microbiome-friendly label, which can be used in the final product. And we believe that in this way, we can, with this analysis, we can, the products that are tested can lead towards a more personalized and skin-friendly cosmetics and therapies. And yeah, with this, I will leave the floor to the next speaker.
Thanks a lot, Paula. There are some questions in the chat. I left some to be answered during the panel. The rest you're free. For all the other participants, some of you did add your questions into the QA. If not, please move them from the chat to the QA box so that we are all centralizing the questions. Thanks a lot, Paula. With that, I would like to introduce our next speaker, Dr. Sebastian Mongiat from DSM, who is a scientist with experience in applied R&D and is known for his co-inventions in applied patents. He's going to talk or walk us through some tips and tricks on the formulation side and from the skin's perspective and microbiome. Sebastian, the screen is yours.
Right, so thank you very much, Radhika. Thank you, Paula, also, and good afternoon, everyone. And very glad to share with you some reflection and tips around the skin microbiota and a way of formulating cosmetic end products appropriately. So now I will share my screen, and Radhika, just let me confirm that you get the appropriate.
Yes, we see the screen.
Okay.
Yes, perfect.
All right.
Thank you.
Good. All right. So microbiome friendly. So here there is, as you all, everybody knows, there is a growing demand on the microbiome friendly certified ingredients, which is also witnessing the increased interest from consumers on the cosmetic, which are more respectful on the skin integrity. So indeed, many brands already developed their communication, such microbiome gentle approach by Dove, which were one of the first by this motto, being gentle to the microbiome starts with being gentle with the skin. Respectissime line also from La Roche-Posay. Here there was a lot of gaining understanding on the microbiome and also the correlation with the physical barrier. And also not only on the cosmetic, also laundry products, and I'm thinking about the Persil, also there, using the surfactant system for clothes cleaning and also respect your skin. So in front of such enthusiasm, we at DSM definitely needed to take up the next step and to provide the right technical guidance in consumer confidence. So naturally, we were proposing not only microbiome-friendly certified ingredients, but also customized microbiome-friendly formula solutions. Here, just a few comments on what stay on the skin. So there are numerous compounds from a beauty care routine still remain on the skin surface after their topical application spreading, and for a certain period of time, despite daily showering. And this is naturally depending on the physical chemical profile of such compound, but also on the cosmetic product used and its location in our body surface area. Beauty end products alter both molecular and bacterial diversity. And seemingly the molecular and the bacterial temporal variability is product, site, and person specific. To illustrate that, I invite you to read the nice paper from Buslimani that I mentioned just below the reference, which is called Impact of Skincare Product and on the Skin Chemistry and the Microbiome Dynamics. Interesting topics, regarding also specific well-known functional ingredient that you can recover in most of the cosmetic products, such as propylene glycol, PEG-derived emulsifier system, and also PPG-derived compounds, which have a certain life shelf on the surface of the skin. In addition to the physical barrier, and the physiological function, the skin is an habitat for commensal bacteria, as Paula was mentioning. Most of that include Staphylococcus, Micrococcus, and Corynebacterium as really the main representative one on the face or arm, legs, whatever. They act also as a barrier against colonization, but potentially pathogenic, but also opportunistic pathogen microbe already present on the skin. So the skin is a dynamic ecosystem. The commensal bacteria are fundamental to the skin physiology and also the immunity. And they metabolize what we call the host proteins and also the lipids, and might also produce bioactive molecules against other microbes. As an example, we can get the sapienic acid from the sebum lipids, and also they can stimulate the host cells itself. This is, I'm thinking about the keratinocyte-derived immune mediator, such as the interleukin-1, and also the induction of what we call the regulatory T cells. So again, here in this scheme, the skin microbiota, the skin integrity, and the immune system connected are working currently together to maintain a beautiful skin appearance. And at DSM, the microbiome approach is mainly driven by really focusing on the corneal care philosophy. This is this holistic view of the epidermal skincare, and also focus on the interaction of such microbiota in the epidermis. And indeed, we have several bioactive in our portfolio that were supporting maintaining such kind of equilibrium. To name few names, I introduce here, for instance, the SYNAP, which was one of our peptides, was the first ingredient that we were evaluating for this impact on the skin microbiota, niacinamide PC, YAC, which are a different grade of hyaluronic acid, pentavitine, our most well-known moisturizer, which is directly targeting the loricrin and the filaggrin at the protein level, stimulation on that, and alpha-fluoralpsebum also there, which is targeting the involucrin for the barrier function on the scalp, for instance. To illustrate slightly more here, there was also an article that we were also issuing in 2018 there, and again with the SYNAP, which were the first peptide here, playing a role on the skin microbiota, and more specifically, as you can see here, on the Staph aureus. This schematic illustration show you really the impact on the plasmin level, which in turn will reduce the inflammation and allow to recover a stronger skin barrier function. I will not dig into all the detail and the specific metabolism pathway there. This is more for my colleagues at DSM, the biologist team, but this was just to illustrate you an example. Another one was on the pentavitine again, here on the scalp skin vitality. Here is the modelization again of the corneocyte. And here you can see we are targeting the filaggrin. So filaggrin, sorry, is an aggregating keratin filaments that help to structure the extracellular lipid matrix in the stratum corneum, which is described here. And as you can see, a loss of filaggrin, but not only, also valuable for loricrin and ceramide, can weaken at the later stage, so the barrier function, and also, by the way, also could entertain a kind of overgrowth of Malassezia furfur in that case, that will contribute to this inflammation and a disbalance of the scalp. So pentavitine here is really breaking such vicious cycle by rebalancing directly the microbiome activity, but also at the same time stimulating what we call this epidermal building block. So again, the outer layer of the skin, so the epidermis is a dynamic structure which can evolve differently following the host physiology, the environment, and also the host lifestyle, what we call the beauty routine that we are using on a daily basis. Here's so skin microbiota, as you can see here, is kind of representing what we call the fourth layer of the skin. You have the epidermis, the dermis, and also what we call the subcutaneous layer. In that respect, we have to integrate the interaction of the microorganism with the skin cells and vice versa. And here I can show you this nice illustration from my colleague Ricardo Sfriso from our biologist group, wrapping up the cause or the consequence of the impairment of this epidermal barrier function, which can lead to skin flaking, which can lead also to increase of this transepidermal water loss, which also will decrease what we call the microbiome diversity, and also here in that case increase the Staph aureus colonization and could also later have this keratinocyte apoptosis, for instance. Among all these interactions, there are proteases secreted by the commensals bacteria, which contribute to this homeostatic bacterial coexistence of the skin, but also proteases from pathogenic bacteria, which act as a virulence factor. So all these postbiotics, let me say like that, produced by the commensal bacteria, and including short fatty chain acid, functional protein, secreted polysaccharide, and many other microbiome fractions contribute to the complexity of such diverse molecular environment, let me say like that. So it is important to strengthen the skin barrier integrity and to optimize the cellular arrangement in this stratum corneum to be in pair with formulating microbiome friendly. So here, maybe it's more focused on these tips. So recommendation by experience, but what we were evaluating since now a couple of times. Very obvious, maybe for certain of you, formulating with surfactant, and especially in rinse-off. So here, try to avoid the sulfate-based surfactants, who will be slightly more harsh to the cell membranes, and prefer to use amino acid, isethionate, glucose-derived, or betaine-based surfactant. What we will not recommend, this is clear, it is formulating with highly cationic compounds for obvious reasons. This is connected to the past of the antibiotic molecule. So all these highly charged quaternized polyquat molecule have to be really precautiously formulated there. What we recommend, which I didn't put on the slide, is also when you are looking for a system to build formulation of your chassis. So whatever it is, a poly or oligosaccharides or polyglycerol-based emulsifier system, these are really well integrated by your skin microbiota and will not disturb that. Preservative system is a huge question. Yeah? So the obvious things is clear that the biocidal preservative should be avoided and prefer bacteriostatic one. And all these preservative boosters, which are totally in line with the mega trends that we are living in the cosmetic industry. So all such as the multifunctional, so the alkandiol, ethyl, exyl glycerin, glycol. So these are helping to get this microbiome-friendly profile. Again, do not hesitate to formulate, with thickening, gelling agent, the water phase and the oil phase of your emulsion or whatever the recipe. And this will also facilitate you to use what we call these mild surface active compounds that are used to build the chassis of your formulation. Here, with the skin product approach, we need to provide these barrier building ingredients, which are naming here that I was introducing to you before. That support the skin natural immunity. So this is healthy skin immune system is a driving life force for both skin microbiome and the skin barrier. So reduction of the transepidermal water loss. This is what I showed you before. Lipids produced by the epidermal cells are mixture of this long chain ceramide, free fatty acid, cholesterol, and so on. So the skin lipids again are these sealing the stratified corneocyte of the stratum corneum in this very well-known brick and mortar skin structure. And in some literature, we could already observe a causal link between a ceramide depletion and an increased Staph aureus population. The best example is the atopic dermatitis, for instance, in the more severe stage. So give a special attention to such ingredient when you design your microbiome-friendly chassis. Normal skin through the life is continually assaulted by what we call this high oxidizing environment, such as the sun exposure, the air pollution, so which are inducing this excess formation of these reactive oxygen species, so which are later oxidizing cellular targets, such as the lipids, the protein, the DNA, and perturbing this barrier function again, and degrading the collagen, which is the major skin structural protein. So keep an eye in formulating with antioxidants in combination with chelators also is helping you also in that sense. But again, association of different ingredients cannot be always predicted, and I think it is really a case by case to get a formulation chassis which will respect your microbiota. There is no universal solution for having a microbiome-friendly solution. It is really a case by case, especially with all the interaction and all the challenge in the formulation. Claim substantiation, different way to claim substantiate a microbiome-friendly formulation. In vivo clinical setup. So this is what we were already engaging with BaseClear. In vitro cultural methods, but also in vitro tissue models. These are the different ways to have your substantiation for a microbiome friendly. Here I give you an example that we were using for this in vitro cultural methods. We were using a standard microbiome here with this specific bunch of bacteria, which are the representative bacteria, whatever it is the face, skin, or the arms and leg skin, there is specificity, there is several models inside that, and also on the scalp, where we have also this modelization. And this was one of the reasons we wanted to have this really, for time and cost efficiency, some evaluation. We have several material and several end product solution we wanted to get as quick as possible. So here in brief, I will show you how we were focusing on 4 distinctive aspects. So the first one, as you can see, is the microbiome quality. That's clear that here we are assessing the total count of these aerobic microbial counts, but also the total count of the yeast and molds, and should be below this 1000 CFU per milliliter. And also the absence of the E. coli, Pseudomonas aeruginosa, Staph aureus, and Candida albicans. The other parameter where we are also focusing is influence of the product on the skin balance, which is represented by the ratio of Staph epidermidis and Staph aureus. You can see that in many literature, in many patents, that the people are focusing when they want to have this modelization of the balance. This is a good indication. And here we are evaluating the ratio simply of the Staph epidermidis versus the Staph aureus. The influence of the product in the co-culture on the bacterial diversity is also very important. So that means that neither an increase nor a decrease of the overall number of the microbial strains should be also respected to get this microbiome-friendly certification. And finally, the influence of the product, direct contact or indirect contact on the growth behavior, where we are evaluating for single bacteria. And here also, after a certain time of exposure, we are always comparing with the placebo on the specific condition of temperature and also with specific nutritive media. Here is just a very simplistic way to assign a number, an index number, which is representative of a grade of microbiome friendliness. Grade 1 here, as you can see, is when the impact triggers a neutral to a slight increase of the evaluated ratio. So that means that here the ratio that you are evaluated should be around 1 or even slightly higher, and that would get a microbiome friendly. When this ratio that was here explaining is slightly decreased to neutral, that means that the value should be between 0.7 and below 1, then we will get a kind of microbiome neutral. And when your ratio is really below 0.7, so that always the ratio with the product and with that, then you can see that here you have what we call a microbiome damaging start effect. Here, just to illustrate, these are a lineup of microbiome-friendly products that we were proposing already. Just maybe due to time, I will not go through. But I can really easily share to you which are the different clear complexion. You have a Flecky shampoo with this mild cleansing surfactant. You have age delay also integrating several ingredients and also a daily SPF, a balanced harmony, which is also a nourishing trend there. And the last one, which is a radiant even skin tone, also featuring some vitamin B3, allantoin, and vitamin C derivative also, which are participating to this skin integrity and the wellness of this microbiota there. Should be the last slide, yes. So Again, if you want to contact, you have the things. I invite you to explore The Secret Life of Skin. This is a website that we were creating where all the people, all scientific community can discuss, share the different, and blog and the articles which are connected on that. So should be the last one. Yeah. So thanks again for your attention. I was just extending, and maybe I will give you now probably to you, Radhika or Rob.
Yeah. Thanks a lot, Sebastian. Also, there were quite a few questions for you. Some
I didn't see, sorry.
I'm glad you didn't see.
Oh, okay.
Some I marked to be discussed for the panel. The rest are free for you. With that, I would like to introduce with pleasure Rob Holland. Rob is a microbiologist with enormous experience with intervention studies and strategies. And he has worked for reputed institutes in the process. And right now, he leads numerous microbiome-related projects at GSK, mainly specialized in respiratory health and oral care. He's going to enlighten us on what is microbiome friendly for oral care products. Rob, all yours.
Thanks, Radhika. Can you hear me okay? Can you see the screen okay? Are my slides okay?
All perfect.
All the technical issues from before are ironed out. Yeah, good evening, good afternoon, good morning, wherever you are. Thanks very much. And thanks very much to the organisers for inviting me to come speak. I've been to a few of these. I've been lucky enough to present at one of them before, and the only thing that ever is slight negative for me is the imposter syndrome I get when I'm in a room full of 170 odd experts and with such broad knowledge on the microbiome. So yeah, thank you for inviting me. Topic of conversation from my side today is just to touch on a little bit what does microbiome friendly mean for oral healthcare. Very much kind of my opinions as opposed to anything formal from a GSK standpoint. And I think most people on the call will be familiar with this, right? You know, we're part of a field with that's growing at a quick rate. You've got a growth rate of 22.5% by 2027, driven in no small part by the increase in science going on in across the microbiome field broadly, but particularly in oral health. Just a quick PubMed search, using oral microbiome as a search tool puts in, brings up the graph on the left showing quite an exponential rise in publications around the oral microbiome. And there's also increasing consumer awareness of the microbiome. Largely, it has to be said around the gut microbiome in a lot of cases, often for the skin microbiome as well. But it's kind of becoming increasingly more of a common term when it comes to, or commonly acknowledged term when it comes to consumer understanding. And a couple of excerpts from a bunch of different news outlets. And I don't know if anyone else in the UK knows who Hugh Fearnley-Whittingstall is, but more recently he's a celebrity chef over here in the UK, and he's actually started putting out cookbooks that talk about cooking for your microbiome and supporting your microbiome. So again, the concept of the microbiome is kind of slowly embedding in the consumer consciousness. And there's also quite a lot of strategic initiatives being made by a lot of different companies like Unilever, Colgate, RB, P&G around building their understanding and building their potential future pipelines around the microbiome. So all this paints quite a strong picture of microbiome being something that is building momentum in the consumer space. This very much oversimplifies it, but I think if we're talking about what microbiome friendly means in an oral health context, there's kind of two things we need really. There's obviously the right product capable of being microbiome friendly, and then there's an understanding of what constitutes a healthy microbiome. And big question when it comes to the oral microbiome is there such a thing as a healthy oral microbiome, a broad healthy microbiome across, or is it more individual than that, as opposed to a kind of a consumer-wide healthy oral microbiome? If we look at series of oral healthcare products. A lot of them you could say would be pretty much not microbiome friendly, driven by the requirements from the consumer, driven by market requirements, and a whole heap of other factors that, you know, so if we look at, say, Polident, which is a denture cleaning cleanser tablet, we've got claims on the pack of, you know, kills 99.99% of bacteria, which, you know, is a requirement for efficacy from a consumer standpoint in terms of denture cleaning. So something that, you know, would not necessarily be considered microbiome friendly, driven by a heap of different ingredients, be they surfactants, be they things that function or can function as antimicrobials. You look at something like mouthwash. Again, we see claims on pack around kills 99.9% of bacteria, so not necessarily speaking to anything that's particularly microbiome friendly per se, driven by the inclusion of things like CPC. And if you look at something like Parodontax, for example, I don't know if you can quite make that out, but we've got claims of removes plaque, and driven again by surfactants and various antimicrobials and abrasives that are included within the formulation. So on the face of it, not necessarily microbiome friendly. And obviously all this is an adjunctive, all of these products are an adjunctive to brushing, which is a physical removal process, which is designed to be a physical removal process, something that our dentists do when we go and visit them, and also something we do, hopefully we all do, twice a day at home as well. If we backtrack a little bit to what constitutes a healthy oral microbiome, as with a lot of microbiome at different sites, it's driven by a whole host of factors, things from, you know, the individual genetics, here on this kind of wheel on the right in blue, intrinsic factors related to the oral environment, so temperature of the oral environment, pH, buffering, salivary buffering capacity, things like that. Host factors, habits, and things like that, things like smoking, things like that, and also extrinsic factors, so socioeconomic status and environmental factors, which the consumer doesn't necessarily have any part of, and all of those interplay and often kind of intertwined in causing quite dramatic differences in individuals' oral microbiome. And I'm not going to touch on it too much in terms of what we're going to talk about now, because we're going to stay kind of more focused on what is a healthy oral microbiome. But we all know that, you know, obviously the microbiome is important from an oral health perspective, is important from a driver of gingivitis and periodontitis. But it's also important to point out that it's also important from a systemic health perspective. So in addition to conditions that would be more considered orally relevant, so bad breath, tooth loss, dental decay, and gum disease, there's also strong links with the oral microbiome in terms of lung conditions, heart disease, strokes, and diabetes. And I've called out one specifically here, which is around the capacity of the oral microbiome to convert ingested nitrate to nitrite and then downstream to nitric oxide. Nitric oxide being an important vasodilator, and dysfunction with nitric oxide signaling has important implications from a point of view of pulmonary hypertension, obesity, cardiovascular disease. And there has been studies that have shown that oral bacterial mouthwashes, for example, can increase blood pressure as a result of that inhibition effect on the oral microbiome. So very much oral microbiome, or the effects of the oral microbiome, can be seen further than just the oral cavity. Worth pointing out as well that after the colon, the oral microbiome is the second most diverse microbiota in the human host, with about 700 or more than 700 bacterial species identified. It has the largest core set of microbes, and I'll come back to that in a little bit, but the main genus found at a large number of sites across a large number of samples is Streptococcus, and there's other key key species and key families that come out as well: Veillonella, Prevotella, Fusobacterium, Porphyromonas, Neisseria. There's a heap that kind of form that core microbiome. But again, coming back to that, so there is that core microbiome, and there's an understanding of what that core microbiome looks like, but there's also quite a lot of diversity among individuals. It's kind of nicely illustrated by this publication by Hall in 2017. So they did a lot of, they took a lot of samples from a number of healthy individuals longitudinally over quite a long period of time, and they identified again that core stable microbial component, but also a variable component, and that's what drives some of the individuality to the microbiome. So really that kind of paints a picture that it is actually quite difficult to pin down a healthy oral microbiome, and that actually really it might be a little bit more individual than that. And a lot of that is driven by the complexity of the oral microbial environment. So look at the diagram on the left, that's teeth. And you've got supragingival plaque, you've got subgingival plaque, and the microbial component of that will be different based on number of factors such as shear stress, mastication forces, so when you chew, and again the shear stress involved in that, and also oxygen gradients and things as well. If you look a little bit broader than that, you've got a number of different environments within the mouth, all with different dominating species and all with a different microbial makeup, and that's quite nicely illustrated in this publication from a year or two ago by Wilbert, where they use classy fish, and that video will behave for me, will hopefully show there's both the diversity of just the microbiome on the tongue, on the tongue dorsum, but also its spatial organization as well. You can see that a little bit better on the left with a little bit more diversity built into that imaging as well. But again, as we've said before, that species composition is different depending on whether you're looking at free bacteria or epithelial bound microbiota, and also is quite different, significantly different across individuals as well. So if we go back to this microbiome-friendly piece, I suppose a lot of what we can understand about what microbiome-friendly might mean comes back to that core microbiome and an understanding of how that microbiome forms, or that plaque or the biofilm on the in the mouth forms. And this is quite a well-established and fairly old kind of understanding of how plaque forms. But if we look at things that we would call oral commensals, or the yellow complex based on the Skransky model of what groups of bacterial species are involved to varying degrees in periodontal disease, things like the Streptococcus and Veillonella, they very much are some of your early colonizers. So some of the bacteria that attach initially to the salivary pellicle and form some of the vice physicochemical bonds, and then some of those initial colonizers of the plaque. If we start to look at things like the orange complex, so some of the bacteria that have links to periodontal disease, Fuso, for example, and for Fusonucleatum and Prevotella, they very much are some of the later colonizers of plaque. Ditto the red complex of bacteria that have very strong association with periodontal disease, so P. gingivalis and Treponema denticola. Again, they're some of the later colonizers of plaque. And if you take something like malodor, so halitosis or bad breath, whatever you want to call it, some of the main players from a point of volatile sulfur compound generation are P. gingivalis, Tannerella, Fusobacterium nucleatum, and again, some of the later colonizers or bridging organisms that are involved in the formation of plaque. So that kind of paints a bit of a picture that immature plaque could be considered healthy, and mature plaque is not, and which is exactly why it's good, as we hope we all do, that you maintain good daily oral hygiene from the point of view of suppressing the the formation of mature plaque, because those are the ones that that form of plaque is more likely to be involved in some of the diseases and some of the conditions that would be associated with poor oral health. So going back again to this microbiome friendly bit, what does that mean from the point of view of how we might go about making microbiome friendly claims? I think it's difficult, would be my opinion. I think your microbiome is complex and it's obviously highly variable. So I think picking what constitutes a healthy microbiome across a population is challenging. Maturity of plaque is important, and removal and control will just naturally always be important to oral healthcare. I think what is starting to happen from the point of view of microbiome type friendly claims, if that's even a phrase, is because we have this understanding of what core species involved in health, and we're beginning to understand the ones that are linked to disease. Whilst microbiome friendly might be a difficult claim to kind of make, the claims around boosting or maintaining good bacteria or reducing bad bacteria, particularly in the context of things like bad breath, are slightly easier to make. And there are products on the market, Zendium from Unilever or Tom's of Maine, which do make those claims around boosting good bacteria and supporting your good bacteria. And so clearly possible, but whether or not we can get to microbiome friendly claims because of some of the things we've talked about, and certainly some of the factors around how oral healthcare products work and their market requirements and consumer requirements will be something that is likely to be a challenge for a while longer in the field. And I think that is me done.
Thanks a lot, Rob.
Back to Radhika.
Thank you. Also, same questions for you in QA. A couple of them I marked for the panel. The rest, you're free. With that, I would like to introduce to you all our last speaker, Dr. Suren Keuros, CEO of Lactobio. Suren has impressive experience to have worked in the senior roles as well as in MT roles for many pharma companies to the records of Novozymes, Panthocare, and Santaris. Lactobio, under the leadership of Suren, has even managed to develop but also commercialize a microbiome-based product. And the talk which Suren is going to show now, or the presentation, has information about that product. Suren, all yours.
Yeah, I will share my screen.
Perfect. It works.
It works.
Correct, it does.
I'll just see if I can share. And can you see my whole screen now in presentation mode?
Yes, we do.
Okay, good. Thank you. Yeah, and Radhika, thank you for inviting me, for giving some insights on what we are doing in Lactobio. I'm very grateful to that. So I'm Søren Keuros and founded Lactobio 5 years ago. And we have worked a lot in the skin microbiome space, trying to develop the good bacteria into skincare products. And I fully agree with the other speakers. It's difficult to define what is microbiome friendly, but at least I will show you some data on what we have done with lactic acid bacteria friendly ingredients. And really, our aim in Lactobio was to develop new skincare products based on microorganisms. And what we see today is that prevalence of skin disease, like many other lifestyle diseases, going up. The diversity of the microbiome, both in the intestines, but also in the skin, is likely going down. A lot of these people, they are quite depressed because you really don't like to show up like this 9-year-old boy with eczema all over. And then also one of the challenges is that most of, for example, acne products, they are quite harsh on the microbiome. So we wanted to find a solution for that, and we have worked with lactic acid bacteria, mainly lactic acid bacteria which are already on the different positive list for dietary supplements, at least here in EU, the QPS list. and these has to be microbiome friendly, depending on how you are defining this. And here in this case story, you can actually see this 9-year-old boy with atopic dermatitis and also staphylococcus infections that our product have improved a lot. He's even smiling here. So in Lactobio, our go-to-market strategy is that we are making finished products. We have made our own branded Back skincare line that we are selling in Scandinavia and also in US now. But we are also doing white label finished products for different cosmetic companies and also other companies. In addition to that, we have developed our own postbiotic ingredients, and this is actually ingredients which are highly bioactive. We have developed a process where we don't heat inactivate the bacteria that we are using, but kill them in a gentle way, if you can say that. We are selling in China these ingredients, but also in different brands in EU and also in Denmark, where we are from. Here you can see our microbiome-friendly products. They don't have any preservation compounds in them. We formulate with oils and lipids, and then it's not necessary, even though for the EU legislation, to have preservatives in the products. We are mainly focusing on problematic skin like acne, eczema, rosacea, and then also on more general daily care. And in all these products, we actually have real life bacteria. They are not growing, but at least they are in the products. And as you will see later, they are in an inactive form. And then when they are applied on the skin, they start growing. The unique concept about Lactobio is that we have made our own proprietary strain collection. We have isolated a lot of strains from humans, healthy humans, fecal samples, vaginal samples, skin samples, breast milk, whatever. And then also we have isolated a lot of lactic acid bacteria from plants and vegetables. And the unique thing about us is that we can actually screen, we can kind of find the most active bacteria, a little like finding a needle in the haystack. And it could be different functionalities, or it could be activity against the Staphylococcus aureus, or the bad or the negative ribotype of Cutibacterium acne. And then we make isolated hit strains. We use different traditional microbiome methods, but also bioinformatics, metabolomics, transcriptomics. And then we also look for patterns in the genes and metabolites using machine learning. We have commercialized the first 4 strains. And these strains, Lactobacillus plantarum, Pediococcus, also Lactobacillus crispatus, has been isolated from different samples. These have mainly been isolated because they are good at killing Staphylococcus aureus, or killing or active against the C. acnes, Candida albicans, also Gardnerella vaginalis. And most of these can be combined in finished products, as we have been doing. And here you can see a lot of diseases, and you're not really allowed to say that when you are using the cosmetic regulation, but you can tell the story around it, and that's actually what we are doing. This is just an example where we have found lactic acid bacteria which are active against Staphylococcus aureus, and especially the ones which are related to atopic dermatitis. And here you see in a spot assay, you see, for example, that we have isolated very good strains from sauerkraut, for example, whereas we have not isolated any from breast milk. So in this way, you could actually find very good strains. And here is then how we actually qualify the heat strains. You can see here, this is on the left side, a Staphylococcus aureus isolated from atopic dermatitis patient, and you can see this grows simply very good. Whereas when we add cell-free supernatant of one of our heat strains, we completely inhibit the activity of this Staphylococcus aureus. Similar things you would also see for MRSA, for example, or vancomycin-resistant enterococcus. Very important is, of course, that there's no toxins or super antigens or virulent factors in the lactic acid bacterial heat strains. But on the other hand, you still want to have some bioactive compounds, for example, like antimicrobial peptides as these plantacine that we have isolated. So this is of course good, and you want to find active compounds which are as specific as possible, just to let the products in the end be more microbiome friendly. And because we are using live bacteria in our formulations, the ingredients has to be microbiome friendly, or at least lactic acid bacterial friendly. And I think it was a very good definition that that we saw today, that microbiome friendly is maybe where you actually increase the good bacteria and then decrease the bad bacteria. And that's actually how we look on this. When we initially started formulating, we killed all our bacteria, lactic acid bacteria, and then we started screening for specific oils and also lipids. Many of these was actually impacting the lactic acid bacteria, and thereby also likely being non-microbiome friendly. Of course, we have isolated bacteria which are, as part of their defense system, also active against other microorganisms. We try really to find the very specific ones which are more active on pathogens, for example, than generally on other skin bacteria, for example. And then also based on this, we actually look, and for the different metabolites, we look on growing our bacteria in such a way so they are still microbiome friendly. As you can see here on the left side, we have a agar plate where we have growth Staphylococcus aureus. Then we have taken an oil formulation, but also a serum cream formulation with the bacteria. You can see very nice clearing zones here, whereas when you take out the lactic acid bacteria, we should see no clearing zones. So this definitely indicates it is the bacteria which have the activity on Staphylococcus aureus. On the other hand, we had to find a solution to get a long shelf life. We would like to have a shelf life of 2 years of our formulated products. And for that reason, we are actually using freeze-dried bacterial crystals. And then what happens is that they are inactive when they are in this lipid oil phase formulation. And then when you apply them on the skin, they are actually activated. And I have just this small video here where a water drop is going over one of these crystals. And what you can see is that the bacteria are activated, and we can also measure that they are starting growing and metabolizing on the skin when they meet the humidity on the skin. And we have made small initial clinical testing, and here, for example, we have made a before and after study of one of our creams with live bacteria. And in 24 patients, we actually saw on this clinical eczema area severity index score that we saw a very nice reduction even after 14 days. Also, when you look on itching and pain, we also saw a huge reduction. And this is likely because the bacteria, the active bacteria here, lactic acid bacteria, they are active against Staphylococcus aureus. And I show you this example again of this 9-year-old boy, which normally use hormone cream and also antibiotic cream, but now they have shifted to one of our products. And here you can also see infection of Staphylococcus aureus. After these 9 days, this boy is even smiling. But what is a good microbiome and what is not a good microbiome? Of course, there is a lot of good results and a lot of knowledge also that the microbiome varies from person to person. But I think most of us is actually can agree that what we actually see here in this case, it is 10 of the patients that we had in this atopic dermatitis study, before after, and before you see a lot of red areas here, and that's actually Staphylococcus. Likely it is Staphylococcus aureus. We don't know yet because it was yes, but we'll look more into this. But definitely Staphylococcus aureus is not good, and then you can actually modify this distribution of bacteria. And what you see working with lactic acid bacterial formulations, skincare formulations, you can actually see you can completely change the distribution of bacteria. And we clearly see reduction of the bad bacteria, the Staphylococcus aureus likely, and then we see some of the good bacteria, Lactobacillus, and likely also we don't know exactly, it is the strains that we actually apply through our cream. So you see the shift, and exactly what microbiome friendly is can be very difficult to define. But I think the definition also we have heard today, that you increase the good bacteria and reduce the bad bacteria could definitely be a way to define this. That was a very short presentation of the work from Lactobio. We are also working with acne and rosacea and other skin areas where we see similar results as I've demonstrated here for atopic dermatitis. Thank you.
Thanks a lot, Søren. I have actually marked quite some questions to be discussed now, and if that's okay, that I'm also going to read them out to the speakers. Some of them, though they are addressed to one speaker, probably are applicable for the others as well. Maybe I'll start with Leah Schmidt's comment, whose question was, is there a relationship between in vitro and in vivo studies, and do we actually confirm the relevance of these in vitro methods? So basically we have now, based on the presentations, we have seen in vitro and in vivo studies, but do we actually go to the level where we are kind of comparing the results and the methods? I don't know who wants to start, or maybe Søren, can I start with you because you had some clinical trials?
Yeah, and the question, I was just reading some of the questions when you were talking, and the question was to actually compare in vitro and vivo.
Correct.
Yeah. Of course, in the end, you need to have the in vivo data. And when you look into very simple things, for example, looking for Staphylococcus aureus reduction, we see direct correlation between what we see in the lab and then actually what we see on the skin. We have also made a lot of swab tests, also looking for specific Staphylococcus aureus strains through PCR examination, and we actually see a very good correlation specifically on issues like that. But of course, it's much more complex We have combined, and we have started also looking on different cytokine profiles now. And there we can see some similarities also, but of course it's much more complex when you look into the in vivo situation. But I think on just sequencing, it will be very important to not only making 16S sequencing, but actually shotgun sequencing, so you hopefully can go down to strain level and make the characterization on that level.
Yeah, perfect. I actually tend to agree with you, but maybe I'll also want to pick with the other speakers. Rob, you deal with a completely different kind of, they are skin and low biomass, but still it has its own challenge. How do you see that link?
Yeah, I mean, I think as with all in vitro to in vivo correlative studies, we probably suffer from the same issues as everyone. Lack of, depending on the model, lack of immune response components and all the other things. I think the big one, often from a oral health point of view, is just that complexity of the mouth that is very, very difficult to replicate. I think a lot of our in vivo studies are the issue with it. In vitro correlation is just often around the site selection and the very difficult to recreate nature of those sites. So I think, but again, I don't think that's necessarily, particularly selective of oral microbiome. I think it's maybe just more difficult when it comes to the oral microbiome.
Yeah. Maybe Sebastian, you have also been in the, let's say, you have had quite some products which are ingredients, but also with a claim on them. How are you seeing them from ingredients perspective, also starting from in vitro, but having a label on a product? Maybe your thoughts on it?
Yeah, maybe. Thank you, Radhika, for that. And thanks also for all the other answers that you were providing, Søren and Rob. I think we do not have to forget, and maybe do not make a false debate. I think we do not have to oppose it, the in vitro with the in vivo, because as you can imagine, we were investing a lot in the 16 RNA shotgun, also with you, BaseClear, also on that. And we were, even if the protocol and the methods are not the same, then we were cross-checking. And I think each of these methods are complementary and maybe also can provide you a specific answer. At least for me, as the formulator point of view, when I'm providing formulation chassis and when I need to ensure that the ingredient that we are providing will not create a disturbance, rather than, because, when you are talking about active ingredients, they are providing an activity on skin, and most of the time also on specific microbes. That's why when we are addressing specific ingredient for the anti-acne and all these type of things, we are addressing that already to a disbalance concern. And I think the rest of the ingredients should not interfere with the activity of your active ingredient. And here really what I was personally involved in for the formulation chassis that I was providing to substantiate all the claim for the in vivo. Also, we have a very cross check and a very close collaboration to provide the minimal impact of the clinical study on that. So I can tell you that sometimes, the modelization is not the same for the in vivo, because there are lots of challenge also to respect the growth of the bacteria in in vitro media, and also with the swab analysis, also the collection of this living bacteria is also something different. But at the end, I can tell you that, yes, for certain specific bacteria, as Søren was mentioning, the Staph aureus, the Staph epidermidis, which are the well-known model, which is already used for many decades, then we were really confirming that we were on the right track with the both study. It's just for a question of rapidity, sometimes that I have a perspective of a formulation recipe, and then you have the perspective of the clinical study, of the claim substantiation of your active compounds, which is something slightly different.
Different, yeah. Thank you. Paula, I know you are more on the measuring side of this game. how do you see as the challenges between in vitro and the in vivo study? And then you're also sitting on some projects wherein you're interpreting.
Yes, I think overall what we said during this webinar is I tend to agree with many of our speakers. I think that, well, from our perspective, the in vitro it's easier to perform because when it comes to single ingredients, because it's simply faster. Of course, it would not be possible to perform in vivo studies testing multiple ingredients that easily. But we believe, on the other hand, in vivo, it's a little bit more strong to test the final formulations because it's possible to target the whole microbiome and not only a specific list of in vitro mock communities. So I think that, in a way, it really depends on which step of the process we are talking about. But I think overall, it's still probably a little bit challenging to draw a final conclusion on this.
Okay, I think it's a fair stance. Maybe a question I'll start with Rob actually. John Burton asked, what might be, in your opinion, the best delivery format for an oral probiotic product? But I would like to extend that question also to the other speakers, not for oral, but at least for in general.
Yeah, I mean, it's a good question. I mean, from an oral point of view, it's quite a challenge, probiotics, right? Because in addition to some of the things I outlined before around market requirements for antibacterial efficacy, these are quite, from a bacterial point of view, hostile products, and they have to be from a stability point of view. So, you know, things like non-aqueous toothpastes are somewhat self-preserving because they haven't got water in them. So I think probiotics in a lot of the normal oral healthcare products that we know and use is quite a challenge. So you're probably looking at delivery. You're probably not looking at toothpaste or mouthwashes or anything like that. You're probably looking more at your standard probiotic delivery methods. Drink sachets or gummies or something like that. Maybe chewing gum, something like that. I think it's very difficult to incorporate into existing products. For those reasons I outlined. I think that's why most people are looking more at prebiotic type routes or paraprobiotics, things like that.
Thank you. Maybe if I can go to Sebastian, also in your presentation, you mentioned something in the lines of textiles. Your thoughts?
What do you mean, that they were extending this, taking
like
care
if
about...
you have a probiotic-based product, what would be the best format?
So again, this is exactly what I was answering at the beginning. I think very well, how you say, I cannot answer to that. There is no universal solution. This is really a case by case, simply because we cannot anticipate, the combination, because there are so many parameters. The intervariability of a person to another person is playing a key role, frankly speaking. Sometimes we are also overstressing on specific ingredients like the preservative system and so on, which are probably not the more, stressful ingredient for the microbiota. They are different things. And then really it was an empirical journey. We were testing a lot. We were learning about the result of the test also on that. And we were building a kind of, let me say, a route of going there. And I was mentioning, polysaccharide. We know that there are lots of polysaccharides also who are playing a role, which is already implemented for many decades. 30 years ago, there are lots of people who were already publishing and providing already product on that area. And at least, there is a common big move at the time being with the cosmetic. Also, we are really into the skin minimalism attitude, and we are using less and less ingredients and even multifunctional ingredients, which are playing different role. And I think this is also the way that as a formulator, we have to follow. Try to reduce also the number, because there are so many interactions. There is already a huge chemical environment on this on this level. And so that's why pretty, how do you say, pretty, yeah, it will be a genius question, I would say.
Paula, do you want to add something in the context of, let's say, if you have a probiotic product and yeah, analysis, it's definitely going to interact. It's definitely going to interrupt the microbiome.
Definitely, but hopefully you can try to trace it back. At least, for example, we try it with qPCR, but I also understand sometimes it's really difficult to deliver the probiotic actively in the specific site. So I also thought with Rob that, for example, gums could be the easiest way to deliver it. I also thought about those device that you put on your teeth to avoid that they scratch. I don't know if you can apply sort of toothpaste or something that can deliver overnight the product. Maybe it's something already developed. But yeah, also the pharmacokinetics and everything, there are so many aspects to take into considerations.
Okay. I'm also looking at the time. So I have one question, but it's actually from Thomas O. I know the answer, probably. I know there is no good and bad, but still I'm tempted to ask. Thomas's question is claim substantiation, which is microbiome friendly, more for marketing purposes or regulatory purposes? Suren, can I start with you?
I think if it's regulatory or marketing, of course, it's something many companies are using now as a marketing tool. But I actually think it's very positive to focus on this, so you actually the consumer can understand a little better what is microbiome. First of all, that's one thing. But also that you actually can use some compounds which are not good for you or your bacteria, and just introducing the consumer to that is very positive. And then for me, it doesn't matter if it's regulatory or if it's a marketing stunt. I think, Radhika, did you leave? Maybe. Okay.
Maybe while she's come back, I'll just add to that, Soren. I think I agree with exactly what you've just said. I think the other thing that I think microbiome friendly fits maybe quite well from a consumer perspective is in the kind of building trend towards naturals. I think you're right. I think consumers need to be educated around what it means and the importance of their microbiome. So I think that's still one thing, but I think the increasing trend towards more natural ingredients, I think that that's a useful place for microbiome friendly things to operate, maybe.
Yeah, and maybe a good advice from me is that you should look more into biofilm, because we know some of these probiotic they can actually inhibit biofilm by co-aggregation to some of these bad bacteria. I think Radhika is back now.
Yeah, my system somehow froze. I don't know why. Just came back.
My technical problems are infectious, I think.
They are. I don't know if we heard from Sebastian or Paula about the microbiome-friendly claim substantiation, unless I missed.
No, I think I will not add more than what was mentioned. I think, yes, I will say rather than marketing or regulatory, I will rather position an efficiency positioning. I think all these things should have a minimum of sense of efficiency. And I think you're totally right, Rob, when you are mentioning there is a time now today where we are living this natural index and so on, and also the skin minimalism. I think this is really the time to be very, again, as a formulator side, we need to be careful when we are designing now a new ingredient also on that. So yeah, I would rather talk about efficiency positioning there.
Yeah, no, just to add on that, I think that's a really important point, because I don't think anyone from a consumer product point of view wants to be pulling the wool over consumers' eyes. I think microbiome friendly as a claim still needs to mean something in terms of efficacy. Yeah. So I completely 100% agree with that.
Yeah, totally right. Yeah.
My stance was more or less the same wordings, that it should benefit the consumer in the end. I don't really mind if it's a marketing trend or a regulatory. It does help me as a user of that product, or it doesn't harm me as a user. Looking at the time, I would like to close off this meeting. Many thanks to all the speakers. Thanks for making time. Also for all the participants who have joined across the ocean, irrespective of time zone. Thanks for being with us. And also thanks to Microbiome Times for co-organizing with us. Just a little bit of a request to the speakers. There are some questions which are unanswered. I don't know if you'll be able to access them after the event. If not, I'm going to send them across to you so that we can also share them with the audience. With that, millions of thanks. Take care, and we'll see each other soon.
Thank you.
Thank you very much.
Bye.
Bye-bye.
All right. Many thanks again for that opportunity.
Thank you.
And take care all. Thanks again.
Yeah. Bye.